All right. Good morning. Welcome to week 4. This is video lesson number 4 for Explorer CT Science. I am still missing you. Who knows if you're missing me, but hopefully this is helpful to you this week. Your goal this week is to complete gene project steps 1 through 6. Step 6 is writing DNA code. You heard J. Craig Venter say that DNA is a software of life. You're the programmer, so you're going to be programming all the genes that are expressed by giving them a gene sequence of A, T, C, and G between 10 and 30 letters long. And then you're going to tell me which chromosome they're on. Of the 10 chromosomes, where are you going to put those genes, like beads on a string? You got 10 strings. And where are you going to put your 21 beads? So completely up to you. You're the programmer. You get to write the code. Before we get there, though, I want to dive into our topic for this week, which is the DNA and then mutations. What happens when mistakes are made in the DNA? And I want to start with Learn Genetics out of the University of Utah and share this perspective. Take a look.
The differences in our genes come about through a natural process called mutation. Mutation is extremely common. As a cell copies its DNA before dividing, it makes occasional typos. Most commonly, a single base is substituted for another. Sometimes a base is deleted, or an extra base is added. The cell is able to repair most of these typos, but each round of cell division introduces a few changes that are not repaired. When DNA changes occur in cells that will give rise to eggs or sperm, they can be passed down to offspring. Each one of us has about 60 new variations that were not present in our parents. People commonly use the terms mutant and mutation to describe something undesirable or broken. But mutation is not always bad. Most DNA changes fall in the large areas of the genome that sit between genes, and they usually have no effect. When variations occur in areas of the genome that code for proteins, they often have consequences. These changes can affect the protein product itself, or they can affect when, where, or how much protein is made. Most of the time, mutation generates variations that are neither good nor bad. Just different.
So mutations are a change in the DNA code. They're a mistake. They're either an addition, a deletion, or a substitution. So I want to dive into what this then looks like. So take out your notes, PowerPoint notes. I got them linked in your folder. You've had them each week. In fact, let's do a quick flip through review where we've been. Started talking about DNA, which is wrapped into chromosomes. The DNA socks, 23 pairs, 46 for human beings that are located inside the nucleus. You unravel it and you get the 2 base pairs, A and T, C and G. Our code is written in that ATCG language, adenine, thymine, guanine, and cytosine. We read about Gregor Mendel, the monk who was a gardener, who was experimenting with pea plants and discovered some principles of genetics. He's called the father of genetics because he found these first principles of dominance and recessive. He experimented with the tall and the short pea plants and showed that recessives hide in the presence of dominant genes and are only shown when you get both recessive genes from the previous generation. We learned about Reginald Punnett, came up with the Punnett square, that when 2 parents each have 2 answers to a genetic question but can only give one, the Punnett square was a means to see the combinations and the proportions of the combinations, the probabilities of what the children would have in their genotypes and phenotypes. All about probability. We then saw that played out with the guinea pig example. We talked about phenotypes, genotypes. We then talked about codominance, where you had 2 different dominant genes that blend together, whether it be black and white feathers, whether it be blue and green glow for the Seahawk glow, red skin, blue skin, purple skin, that concept of codominance. There's a shared dominance between the genes. Then we talked about genes on chromosomes. You're going to be writing code for DNA this week and then putting it on chromosomes. You get to choose the sequence. It has to be unique, but you get to choose the sequence, and then you get to choose which chromosome you put it on. Chromosome 1, 2, 3, 4, 5, 6, 7, 8, 9, or 10. You could put 2 genes on each chromosome for the 20. You can put 1 gene on one, 3 on the other. It's completely up to you, because again, those genes are like beads on a string, and that chromosome is the string. We talked about how cells make proteins. Copy, send, build. The DNA is copied into messenger RNA. It's sent to the ribosome. Ribosome then builds it. You saw the biome video, the video game that we made to show this, and gene edit cancer. I'm going to be talking about that in a second. So we went through that last week. Here is what we're doing this week. We're looking at mutations. So we look at this original sequence of GGGAAA. A substitution would be GGTAAA. There is a substituted base pair for the original. A deletion is when you just lose that base pair, GGAAA. So one is deleted, it's lost. Then there's an addition, GGGCAAA. So in each of those cases, it changes the sequence, and the sequence has meaning, just like letters that make words, that make sentences in a book. If you change letters, you add a letter, delete a letter, substitute a letter. Sometimes it doesn't make a difference in the meaning, but sometimes it does. If I take the word dot and I substitute the letter T at the beginning, now I've got a tot. Or if I substitute that letter into bot, well, a dot is different than a bot. And so the change in the letter changes the meaning. And whenever you have a change in the sequence, it may mean a change in the meaning of the protein that you build. It may not. You just don't know. It depends on where it happens and the sequence of the meaning. So we're going to see more about mutations here in a second. Now, I also wanted to finish off with a pedigree. A pedigree then tracks those genes that are passed from one generation to the next. And so many of you have probably seen a pedigree of genes that have been tracked through your family, or at least seen an example of one. It's just a way to track the inheritance of a family. We're also going to be talking about karyotyping. A karyotype is kind of a map of the chromosomes that you have, and then lining up those chromosomes, because again, DNA forms the genes, and the genes are beads on a string, and those genes are on the chromosomes. And so we're going to get a different look at that today as well. And that is it for our gene notes. We'll come back to that later. What I now want to do is now use the biome video again to show the impact of mutations. So take a look. And again, since we don't have the game or the cart, I'm just going to show you in the middle of the programming what we did and why we did it. So again, you're the organoid. You're flying inside the nucleus through the nuclear pore. As you go in, you'll see the chromosomes. Those chromosomes will light up green if they don't have mutations in them. They light up red if they do have mutations in them, changes in the DNA. So as we go through, we find chromosome 12, and the gene we were looking at was specific to cancer. And again, if we unravel the chromosome, and then I look at those black areas, the area is not really black, but it represents a mistake. So look up here. I should have an A with a T, but that's a T. I should have a G with a C, an A with a T, a G with that C, an A with that T. Should have a G with this C. And yet those are all mistakes. And so in the game, which is fictional right now, hopefully soon it'll be reality, that we can actually gene edit mistakes that cause diseases like cancer. But then we go back through, we match them up, and we fix those mutations. And after this, we then take it to the ribosome and we build the protein. By the way, if we don't change those, then that bad sequence often leads to making the wrong protein, or a bad form, a corrupt form of the protein, which then can lead to the cancer as well. We'll come back to that in a little bit. So That said, let's talk a little gene project. Up until now, you've been dealing with the genes, the dominant, the recessive, the genotype, phenotype of mom and dad and the child. Well, again, this week, it's about as easy as it gets. Step 6, assign a DNA sequence and chromosome location for the dominant alleles for each of the genes in your species. Assume there are 10 different chromosomes for your species. So look at my example. I used a sequence of ATCG between 10 and 30 letters long to represent the gene for wings. And again, I don't have to do both genes on each one, just the one that's expressed. So if the dominant's expressed, write the code for the dominant gene. If it's just the recessives that are expressed, they're homozygous recessive, they have 2 little t's or mammoth tusks, then just give me the sequence for little t. You're just writing one sequence for each trait that is expressed. Then over here, chromosome location, I just put that gene for wings on chromosome number 1. I might do 1, 1, 2, 2, 3, 3, 4, 4, just to space it out. Or you could do something different. So again, let's say we're talking about tusks. And I say, all right, I'm going to make the genetic sequence for tusks. Well, the only thing I can't do is use the exact same sequence as wings, because that's now the code for wings. So I could do CA, GG, CA, TA, CG, GG, AC. And as long as it's at least one letter different, that could now be my genetic sequence that codes for tusks. And I could put that on chromosome number 1. Or I could do 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, and then go back through and do 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 and spread it out. Again, completely up to you. You're the programmer. You get to write the code. It's the software of life. Let's do another one. Let's say I'm writing code for number 2, and let's say code number 2 is for fins. Okay, I ended up with shark fins. Could I actually do something like that? Sure. Is that a sequence of A, T, C, or G between 10 and 30 letters long? Yep. Where could I put it? Doesn't matter. Beads on a string. You've got 21 beads, 20 dominant recessive, 1 that is codominant. You're just telling me which 10 strings you're putting those on. So I could do chromosome number 3 for that. Could I come down here for the next one and do a code like GC, 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 all the way through? I sure could. And maybe that is coding for stripes. And I put that on chromosome number 4. So again, the only rules are between 10 and 30 nucleic acids long, A, T, C, and G, and you just can't use the same sequence you've used before. In real life, we're talking hundreds and usually thousands of nucleic acids that spell a gene. In fact, what I want to do now is show you what happens when one of those genes that spells something like fur color gets broken. So I'm going to now take you to Howard Hughes Medical Institute, Making of the Fittest. I want to show you a clip showing what happens when a gene gets broken and changes a trait. Take a look.
Across the American Southwest, golden deserts dotted by cacti and brush stretch for miles. Yet here in New Mexico's Valley of Fire, the landscape changes dramatically. Patches of black rock interrupt the sand, remnants of volcanic eruptions that occurred about 1,000 years ago. The eruption spewed a river of lava more than 40 miles long across the desert. As the molten rock cooled, it darkened, leaving any creature dependent upon camouflage in serious trouble. In the complex battle of life, one of the constant struggles between seeing and not being seen, the evolutionary game of hide and seek. And we've come here to the Valley of Fire in New Mexico a battlefield, to find one of the tiniest soldiers and what it can teach us about how evolution works. On the desert sands, the rock pocket mouse blends in perfectly, its light-colored fur concealing it from predators. But on dark lava, the same fur makes the mouse stand out, attracting the many creatures that see it as food.
These mice are the Snickers bar of the desert. They're eaten by foxes and coyotes and rattlesnakes, and certainly by owls, and maybe even occasionally hawks. And most of those predators are visual predators.
So what happened to the pocket mice that found themselves on this new terrain? When I accompany biologist Michael Nachman onto the lava, it doesn't take long to find out.
Oh, this one's closed.
Nachman has been collecting mice unharmed in traps. And it's a dark one.
It is, yeah.
Now, are most of the ones you find up here dark?
Almost all of them.
Not only have the mice here evolved to be as dark as the rock, the color change has occurred precisely where it will conceal them from hunters.
A bit of a white underbelly, too. That's right. All of the dark ones here and on other lava flows have a white underbelly, and presumably there's no selection for dark on the belly because predators are coming from above.
Left to themselves, the mice show no preference for light or dark rocks. It's the predators that have made the difference.
The change in color over evolutionary time in the population is driven by predators weeding out the mice that don't match their background.
But how did the dark mice arise in the first place?
When a black mouse appears in a light population of mice, that is usually going to be due to a new mutation. And those are random and rare events.
To fully understand the pocket mouse transformation, Nachman moves from the lava to the lab. He and his team extract DNA from light and dark mice taken from one desert region. The aim? To find one or more genetic mutations that cause dark coloration. A mutation is a change in the chemical letters that make up our genes. It's a copying error that may occur when our cells divide. Mutation seems to mean that something bad has happened. Well, mutations are neither good or bad. Whether they are favored or whether they are rejected or whether they're just neutral depends upon the conditions an organism finds itself. So for the pocket mouse, a mutation that causes a mouse to turn black, that is good if you're living on black rock. It's bad if you're living out in the sandy desert.
The light mice are all on the bottom, here.
Fur color is a trait controlled by many genes.
Turns out.
To figure out how dark mice evolved, Nachman focuses on how these genes differ in dark and light mice. One by one, the genes prove identical, but at last, something does turn up. The difference between dark and light mice boils down to a difference of 4 chemical letters in a gene called MC1R. Because the gene controls the amount of dark pigment in a mouse's hair follicles, a mouse with these mutations grows dark fur, which gives it an advantage on a dark background. But still, that's one mouse. How would its dark fur spread to a whole population?
This lava flow is about 1,000 years old. And so you might wonder, has there been enough time? It's only been 1,000 years. It's a very short period of time for a new mutation to come along and spread so that all of the mice on this lava flow are black, because really they all are.
Indeed, such a rapid spread of a mutation may seem unlikely until you do the math. And the reason is that while only one new mouse born in 100,000 may be black, hundreds of thousands of mice are born in any given year. And then those mice that are black have enough advantage that their babies do better and they have more offspring and their offspring have more offspring. And just about a 5% advantage compounded year in and year out can very quickly turn the whole population black, as we see today. If dark color gives mice a 1% competitive advantage, and you start with 1% of the population being dark, in about 1,000 years, 95% of the mice will be dark. If instead, the dark color gives them a 10% advantage, then it only takes 100 years. Thanks to Nachman's mice, science has an example of evolution crystal clear in every detail.
What's exciting about this is that we have a system that's very simple ecologically. You have dark rocks and you have light rocks, and you have dark mice and light mice. It couldn't be simpler.
We know who the predators are, what the selective force is. We know precisely the genetic basis of what makes the mice have an advantage or a disadvantage depending upon where they live. All the pieces are finally together. It's a perfect illustration of Darwin's process of natural selection. In fact, it's more than that. For Nachman's mice also counter a common misconception that evolution is a random process. Well, there is one random component, and that's the process of mutation. Mutations occur at random throughout our DNA. Every new organism is born with a new set of mutations. But while mutation is random, natural selection is not. Natural selection sorts out the winners and losers.
And that's what we're going to be working on the following week, is how mutations then affect natural selection, survival of the fittest, and how traits then can select for or against a species' survival. So that is a to be continued, but we begin to see the role of mutations in changing genetic information, breaking genes, corrupting information, changing the color from brown to black, which actually gave those mice a selective advantage. But in most cases, it doesn't. In fact, in most cases, it can cause harm when it is effectual. When it does something, it usually does harm. And one of those harms it can do is cancer. So one of the other explorations I'd like you to think about doing this week, it's optional, but I'd like you to think about watching episode 4 in The Secret of Life, the PBS series on DNA, which is all about curing cancer, which speaks to mutations and then our genetic research and how what we are learning could eventually lead to a cure for cancer, whether that be CRISPR, gene editing. So I want to give you a little bit of a history of that. So take a look. This is just the beginnings of it.
I'll tell you a story. I won't put a name to it. But my wife once had to our house a very brilliant scientist who sat through dinner which she had prepared, saying nothing. And when she finally said to him, "Aren't you going to say something before you go?" He said, "Look, I don't have to say anything. I'm going to cure cancer."
Curing cancer once seemed like an impossible dream, but some scientists now believe they hold the key: DNA.
There are many very serious problems facing humans, among them poverty and ignorance. And about these problems, we can do very little. But cancer is a problem that we actually can do something about, and so I work on cancer.
With cancer touching so many lives, this is science for the highest stakes.
We had been working on something that to me was genetic markers and chromosomes and genes and physical mapping and sequencing, and suddenly it was people's lives and people's families.
Bud Romine was told he was about to die of cancer. But today he is alive thanks to a new class of drug created by DNA scientists. It holds the promise of transforming cancer treatment.
That stuff kept me alive. It's keeping me alive right now. There's no question in my mind. Not a bit. Gives me a little long-range view. I bought a new pickup. We bought a newer place down in California. Now, is that optimism or what?
This is the story of a small group of researchers who are developing radical plans to cure the disease we fear most. They're tracing every cancer back to its origins, back to its DNA.
Those are tumors. Those are tumors created from cancerous sequences of DNA, mutated sequences of DNA that created problems in the protein synthesis that then created problems with cells that had uncontrolled growth, tumor growth, that then led to those tumors. Now, some of this may look a little familiar. We've seen pieces of it. We haven't seen the whole story. So what would be perfect this week is if you watched the whole thing, saw the whole story as it all fits together, because that's what we were going to do when we were going to be together this spring. We're going to put all the pieces together. So I'm hoping that you'll watch this and put the pieces together and see how it all works together. And then cure cancer. I'd like you to cure cancer. Okay, so as we come back then to Schoology for the week, you will have my video lesson. Hopefully you're watching this Monday, Tuesday. Then as you work your way up, you just saw the making of the fittest. You can watch the end if you'd like. We saw the connections we needed. There are gizmos of the week. You can do the gizmo of the week, which is human karyotyping, which is actually looking at how do we organize all the chromosomes for the human genome. It's awesome. You play a matching game. Whoever does this first and gets it right, let me know. I want to know who's first on this one. The bonus gizmo is doing some genetic engineering. That's always fun. You're doing genetic engineering in the Gene Project. So this is another example of changing genes, changing sequences, hopefully to get beneficial adaptations and traits. Here's the link for curing cancer. You just saw the intro to. You can go back and watch the video on what's a mutation from Learn Genetics at the University of Utah. I put the Gene Project and notes back on here just in case you need them. So as far as what's required, step 6, keep building your Gene Project. As far as enrichment material, you can see the gizmos, the links, seeing the piece on curing cancer. I want you to have a great week. I want you to learn a lot this week. And so I've tried to package it well together. Also, if you are not getting the video for whatever reason, I'm also establishing a YouTube channel. Even if you are getting the video, go look at the YouTube channel, put a whole bunch of different videos on there just for fun. So when you get bored, you can go watch fun stuff. So I think that may be all I have for you today. Again, miss you. I will see you at the Google Meet this week. Google Meet's going to be a little different this week in that we're going to be all together. So it will be from 12 to 1, and we will start at 12 o'clock all together. I'll send you the link, and then you will join us, ask any questions, share how you're doing, and then we will go from there. Again, have a great week, and I will see you guys soon.